Hello everyone, this video is about cryopreservation of embryo and organs. So first of all, what is cryopreservation? It is a process that preserves organelles, cells, tissue, embryo or any other biological materials by cooling the sample to sub-zero temperature, usually below minus 150 degrees Celsius. The temperature may range from moderately hypothermic conditions to cryogenic temperatures. In 1949, bull semen was cryopreserved. This led to a much wider use of cryopreservation today, with many organs, tissues, and cells are stored at low temperature. Human sperm, egg, and embryo are routinely stored in fertility research and treatments through this process. Tissue and blood of the body can be removed, frozen, stored, and then later reintroduced into the body when needed. One particular use of this technology is done in bone marrow transplant. Cryosurgery. It is a type of surgery that involves the use of extreme cold to destroy abnormal tissues such as tumors. The surgery most often involves the use of liquid nitrogen, although carbon dioxide and argon can be used. Plant seeds or shoot may be cryopreserved for conservation purpose. Embryo and organs cryopreservation. This particular technique been used for many species of animals, starting with the mouse in 1972 to the goat in 1976 and the human in 1983. In fact, this type of procedure has been so successful throughout many species of animal kingdom. The only species that has still been unable to undergo this procedure with success is swine, that is pig. So many animals from all different species have been born through this process of embryo cryopreservation. The steps involved in the cryopreservation are: first one, selection of materials, addition of cryoprotectants, freezing, storage in liquid nitrogen, thawing, washing and reculturing, measurement of viability and regeneration of plants. The first one is the selection of materials. Few important criteria should be followed while selecting the biological materials such as volume, density, pH, morphology, and without any damage. The second one is the addition of cryoprotectant. Cryoprotectant agents such as glycerol FBS, that is fetal bovine serum, salts, sugars, glycols are added to the sample as it reduces the freezing point of the medium and also allow slower cooling rate, which reduces the risk of crystallization. The next one is the freezing. Different methods of freezing are applied in this method of cryopreservation to protect the cells from damage and cell death by the exposure to the warm solutions of cryoprotective agents. And there are two processes in freezing. The first one is the slow rate freezing, and the second one is the vitrification, that is super rapid freezing. Slow rate freezing. It avoids ice formation inside the cell by making water leave the cell and ice still forms outside the cell. But in vitrification, it avoids overall ice formation inside and outside the cell. Next is the storage in liquid nitrogen. The cryopreserved samples are stored in extreme cold or minus 80 degrees Celsius in a freezer for at least 5 to 24 hours before transferring it to the storage vessel. Next one is thawing. It is a process of warming the biological samples in order to control the rate of cooling and prevent the cell damage caused by crystallization. Next is washing and reculturing. The preserved materials is washed to remove the cryoprotectant. Next one is the measurement of viability. Due to the storage stress, there is a possibility of cell death. The presence of viability can be seen in most cases. The next is the regeneration in plants. After the non-viable seeds are cultured on a specific medium, Suitable environmental conditions are maintained. Applications of cryopreservation. Preservation of cells and tissues for the purpose of long term storage. Cryosurgery, a minimally invasive approach for the destruction of unhealthy tissues. Next is lyophilization, that is, freeze drying of pharmaceuticals and the study of cold adaptation of plants and animals. Thank you.